we really are coming at this from a very molecular and cellular level, what's happening in the brain with chronic alcohol exposure, and also what are the factors in the brain that might contribute to a predisposition to drink excessively.
Welcome to Collaborative Endeavors, a podcast about how scientists from different areas of research come together to tackle big health challenges, leading to better therapies and healthier communities. Dr. Amy Losick is an associate professor in UIC's Department of Psychiatry with a joint appointment in anatomy and cell biology. Her lab is interested in understanding the biological foundation of alcohol and drug addiction using molecular, cellular, genetic, and behavioral approaches in rodents. They also study neurobiological differences between men and women as they pertain to alcohol use disorder, what many of us refer to as alcoholism. In 2018, Dr. Losick was awarded a CCTS pilot grant for a project examining a new therapeutic approach to reduce binge drinking in women. Treatment options for alcohol use disorder are limited across the board, but this is especially true for women who are often underrepresented in clinical studies. Dr. Losick spoke about her efforts to understand the mechanisms of binge drinking behaviors in women, as well as the important role her drug discovery collaborators play in bringing the basic science of addiction a step closer to a usable form of therapy.
My background, actually, my training is in molecular and cell biology. So I got my PhD at Cornell University in New York City at the Weill Graduate School of Medical Sciences. My primary training was in molecular biology. And so I actually wasn't doing neuroscience. I was doing cancer research. So I was interested in signaling pathways involved in cancer. And so that sort of, gave me a really nice molecular and cellular background for the studies that I do now. And then in 2003, I actually switched to neuroscience because there were a lot of open areas in neuroscience, and especially molecular and cellular neuroscience. I guess my main fascination with that was, how do you manipulate a specific gene in a specific brain region and cause a change in behavior? And so that to me was a really interesting question, and especially related to addiction. And sometimes in society we still think of addiction as something that has to do with like a moral failure or some kind of lack of willpower, for instance. But really, addiction is, based on biology, and it's based on both genetic and environmental factors. And I just thought the question of, okay, what if you manipulate a specific gene or a specific signaling pathway in the brain, how does this contribute to behaviors related to drug addiction?
A little over 5% of the population suffers from an alcohol use disorder, which is basically defined as the inability to quit drinking, drinking to excess, or drinking despite negative consequences. Binge drinking is a particularly prevalent form of alcohol use that leads to serious adverse health outcomes for women, including increased risk of breast cancer, organ damage, and mental illness.
So alcohol drinking, excessive drinking, or even sometimes moderate levels of drinking can lead to increased risk of breast cancer. There's an increased level of cognitive impairment with alcohol use in women, and also liver damage and cardiac outcomes. So it's important to study women in this, and what's the biology that's driving alcohol drinking in women versus men, because it may be slightly different biology that's driving these differences in drinking. We're really a basic science lab doing research on a clinical question, but we try to lean in a translational direction so that we can find new drug targets for treatment of alcohol use disorder. And so we have a number of different projects, and one of the projects that we're working on has to do with looking at sex differences in alcohol drinking and alcohol use disorder. And one of the things we know from the clinical literature is that women are more likely to transition from sort of casual drinking to actually having an alcohol problem. And women are also more sensitive to the detrimental effects of alcohol.
Dr. Lasik went on to describe the research study that laid the foundation for her CCTS pilot project.
We work with mice, and we started out this project first just comparing, what happens between males and female mice when you look at it in a binge drinking test. So they're getting enough alcohol that they're showing the pharmacological effects of alcohol. So this is important because we want them to be drinking in a sense the way that humans would drink, where they would be binge drinking and drinking enough to become intoxicated. So when we compared the males and the females, we found that the female mice actually drank more than the males, and this is consistent with what's been out there in the literature for a while, but it was sort of the first time we had done it with this specific binge drinking test. And then we decided, let's figure out why the females are drinking more than the males. And so one of the reasons they could be drinking more is because of differences in their hormones, and specifically estrogen and progesterone. And so what we did is we removed the ovaries from the mice, and then we looked at their drinking behavior. And when we compared the females without ovaries to the males and to the gonadally intact females, they actually drank less. So they drank as much as the males, not at the high levels that females with ovaries drank. So that suggested to us that the hormones are involved in alcohol binge drinking in females. So that was sort of the basis for trying to understand the underlying biology of this. And we did a couple of different things. One thing we did is we looked in the reward region of the brain, one of the areas called the ventral tegmental area. And this is where the dopamine neurons are located. And dopamine neurons actually are thought to be involved in the rewarding and reinforcing properties of drugs of abuse. And we saw that there are estrogen receptors in the dopamine neurons in the ventral tegmental area, actually in both males and females. So both sexes actually make estrogen receptors in their brain and are responsive to estrogen. But the interesting thing was when we knocked down a specific estrogen receptor known as ER alpha, we actually reduced drinking in the females, but not in the males. So even though both the females and the males make these estrogen receptors in their ventral tegmental area, we only had an effect of reducing the receptors in the females. So again, this was sort of a really interesting finding that these estrogen receptors may only be in its region active in the females. We've also done some further studies where we found that alcohol can stimulate the activity of these dopamine neurons, and when estrogen levels are high, the activity of these neurons is higher in response to alcohol. So again, estrogen is sort of implicated in this response of the dopamine system. This kind of gave us an idea of sort of where in the brain this might be occurring and mechanisms related to dopamine. But in terms of a translational aspect, we were really interested in determining, could we treat females with some kind of drug that blocked estrogen receptors and see an effect on alcohol drinking?
Let's take a step back for a minute. Researchers who study the molecular biology of a disease, like Amy Lasik, typically focus on a protein and then perform genetic manipulations to increase or decrease its expression. In turn, this can reduce the disease condition in their model. While this is an important step in validating a target or pathway, these techniques cannot be used to create drug therapies. Developing a compound that might mimic the results of a genetic deletion is an entirely different skill set. And even when investigators properly orient their research toward developing a new therapy, the process of translating the basic science of a disease to a clinical intervention is so daunting that it is appropriately referred to as the Valley of Death. To overcome this challenge, Dr. Lasik's team partnered with Dr. Gregory Thatcher, currently professor of pharmacology and toxicology at the University of Arizona's College of Pharmacy. In his former position at UIC, Dr. Thatcher founded UICentre, a campus-wide disease-agnostic drug discovery unit focused on small molecule drug discovery. The center's collaborative organizational model has evolved to bring together experts with varied skill sets, including project management, bioassay development, high-throughput screening, and drug metabolism and pharmacokinetic studies.
I think the reason I got connected with Dr. Thatcher was because I think I gave a talk in the College of Pharmacy on our work, alcohol use disorder and estrogen. That was sort of the initial connection. And then, we had this idea that we should collaborate because he was very interested in estrogen receptors in the brain and designing compounds that actually could get into the brain and work. So that was sort of the initial connection. And then we just decided, well, this would be a perfect opportunity to collaborate because we had this test set up. We had some evidence that estrogen was involved in binge drinking in females. And so we could use his compounds to do these experiments to see if we could reduce binge drinking. So that was sort of, it was a nice, perfect intersection of our strengths. My behavioral and molecular neuroscience and his medicinal chemistry background and interest in estrogen receptors in the brain. He has developed what are called selective estrogen receptor degraders. And those are drugs that actually can lead to the downregulation of estrogen receptors. And his primary reason for developing these originally was for breast cancer treatment. One of the drugs that's out there that's FDA approved is for metastatic breast cancer is a selective estrogen receptor degrader, abbreviated SERD, and it's called fulvestrant. And this is typically used to treat postmenopausal women with metastatic breast cancer. But this drug doesn't really get into the brain very well. And so for our purposes, we couldn't really think of using fulvestrant as sort of an orally bioavailable drug to reduce binge drinking in females. So Greg Thatcher's group actually developed a SERD that is blood-brain barrier penetrant, so it can get into the brain. And so we decided to do a series of experiments where we treated female mice with the selective estrogen receptor degrader that he developed, the brain penetrant degrader, and then tested the binge drinking that I was talking about earlier in females. So the interesting thing is we were able to reduce binge drinking after a few treatments with the SERD in the female mice. This suggests that orally bioavailable and brain-penetrant selective estrogen receptor degrader can potentially have some therapeutic impact.
While Dr. Thatcher was not available to contribute to this episode, I was able to speak with UICentre's Assistant Director, Dr. Manal Benaisa. She elaborated on Dr. Thatcher's contributions to Dr. Lassik's project, how drug repurposing works, and the role of drug discovery units like UICentre when it comes to the potential identification and development of new pharmaceutical therapies.
My name is Manal Benaisa. I'm Assistant Director at UICentre for Drug Discovery, and I'm also Research Assistant Professor at the Department of Pharmaceutical Science. My work through UICentre, I'm involved in the first stage of drug discovery from early stage to late stage. So I do assay development, screening, and later stage mechanism of action or proof of concept study for in vivo validation. I'm not specialized in addiction, but when Amy Lasik approached us as UICentre, apparently she discovered a novel target for addiction, especially in female. Treating with estrogen increased the behavior of binge drinking in female. And when she knocked down ER receptor in the brain, that behavior decreased a lot. So because of at UICentre, and especially Dr. Thatcher, he has over 20 years experience in developing endocrine therapy for breast cancer. So we thought at UICentre to repurpose this class of compound to try to treat addiction and specifically the binge drinking behavior in female. So we have different class of compounds at UICentre that were developed by Dr. Thatcher. He has SERMs and SERDs. They made it to clinical trial. So SERMs are selective estrogen modulator and SERDs are selective estrogen degrader. So we can degrade ER. And the good thing with this class of compounds are brain bioavailable. It means we can use them, other than cancer, we can use them to target ER in the brain. And because Amy Lezik discovered this novel target, so we wanted to see if potentially we can repurpose this class of drugs to treat addiction in females specifically. So usually the PI, they know their project very well, but they are at different stages. They are early stage or more later stage with more validation, and they know more about the target. They know how they do their assays. But to discover the drug is another thing. So usually our collaborator to come to us with a good assay, but that assay is not scalable, for example, to do high-throughput screening to identify novel drugs. So we can assist them to look at their assay, to look at their target, if it's druggable or not, if we can develop an assay, if we can go to the next step to do screening. We can help them on doing medicinal chemistry to optimize the compounds that were identified from screening. Also, even we can help them at the later stage of preclinical validations because we have a medchem component at UICentre, so we can look at pharmacokinetic of drugs, if they are druggable or not, if they can be available when we treat the animal. So we can help at different stages of drug discovery.
Dr. Benaissa also stressed the importance of considering sex differences in the development of drugs. Research has shown that biological differences between men and women, that is, differences due to sex chromosomes or sex hormones, may contribute to variations seen in the safety and efficacy of drugs, biologics, and medical devices.
I think gender difference or sex difference are usually omitted from clinical trials and try to recognize that there is difference in many diseases, like, for example, in Alzheimer's disease, you have more female, more women that will develop Alzheimer's disease. It's important to try to consider the sex difference in clinical trial to remove the bias during a validation of drugs.
While the use of SERDs to address binge drinking behavior shows promise, an intervention involving the manipulation of estrogen in the brain is not without its risks. Dr. Lassick explained some of the drawbacks to this approach, as well as potential new areas of exploration.
There are complications associated with any drug, and it's important to investigate whether this would have any other effects on females, given what we know about estrogen receptor blocking drugs in premenopausal females. If you give, for instance, tamoxifen to women, there are psychiatric effects of that, so people can become more depressed or anxious when they're on tamoxifen, because estrogen receptors play an important role in anxiety and depression as well. And so you want to be careful, maybe not directly targeting those estrogen receptors, but maybe targeting something in the pathway that perhaps is more specific to the response to alcohol. The estrogen receptors interact with other proteins in the cell, and again, this might be involved in that response to alcohol. If we can block either the estrogen receptors or a protein that interacts with estrogen receptors, maybe we can decrease drinking and decrease that enhanced sensitivity to alcohol. So it's a way of coming at it from a different question. You know, it's sort of like from the side, like, okay, what are the other interacting proteins? One of the things that the prediction from this is that at different phases of the menstrual cycle in premenopausal women, you would have perhaps a higher propensity to drink more alcohol or binge drink. And there is some data out there that suggests that women in their follicular phase, when estrogen levels before ovulation, when estrogen levels are rising, that they consume more alcohol. So I think there is a translational aspect to this. It's not that necessarily we want to start giving women like estrogen receptor blockers, to reduce their binge drinking, but that an awareness of, okay, well, I'm at this specific phase of my menstrual cycle, maybe I'm at higher risk. Maybe that awareness will help, women think, okay, maybe I can try not to drink as much, try not to let it get out of control. So there is a little bit of an aspect of that. The other thing is that, in terms of treatment, maybe stabilizing the hormonal fluctuations in some way with the therapy, like, basically estrogen replacement or something like that. Maybe there are some other things we could look at in humans, like, for instance, in human blood samples, or in looking at like genetic variations in estrogen receptors that might be associated with high levels of drinking. And so that would be, an area that might be really interesting to pursue, is really look at genetic variation, because maybe that would be involved. And I think because they have the samples from human patients. So I think that would be really interesting to do in the future. For me, it's the idea of team science it's really important. I've always loved being part of a team where people bring in different areas of expertise, and I participate in many ways in different teams. So I'm part of our Center for Alcohol Research and Epigenetics here at UIC, and what we're doing is trying to understand epigenetic mechanisms that are involved in basically depression and anxiety during withdrawal from alcohol as a way to target that to prevent relapse to drinking. And we bring in a lot of different expertise. I mean, we have the molecular and cellular expertise, but the behavioral expertise, and then, eventually, perhaps drug discovery once we identify new targets that can be where we can develop drugs based on those targets. And I'm continuing my focus on the basic science, still collaborating with people that do things that I can't do, like the electrophysiology, which is a whole different area of neuroscience. I guess, just in terms of like the area of studying alcohol use disorder, I think it's just really important to think about, how can we understand this disorder, this brain disorder, from the basic biology of it, and really try to find new targets to help people. So, that sort of is motivation really behind everything we do.
Collaborative Endeavors is produced by me, Lauren Rieger, on behalf of the Center for Clinical and Translational Science, aka the CCTS, at the University of Illinois at Chicago. To learn more about the research discussed in this episode, visit the links in our show notes. The CCTS is supported by the National Institutes of Health's National Center for Advancing Translational Science through their Clinical and Translational Science Award. Opinions expressed by the guests of the show are their own and do not necessarily represent the views of myself, the CCTS, or our funding agencies. You can find more episodes of Collaborative Endeavors on Apple Podcasts, Spotify, Amazon Music, and Google Podcasts. New episodes drop monthly. If you would like to see your interdisciplinary team featured on the podcast, reach out to me via the email address in the show notes. To learn more about how you can get involved in health research, visit ccts.uic.edu or follow us on Twitter @UICCCTS.